Hello everyone. Here we are in another session, diving into the world of rejuvenating our DNA, the anti-aging technologies. This is the fourth video of the course, and today's topic is the transformation in plain words, introducing DNA inside bacteria. As you can see here, inserting DNA inside bacteria. Easy, right? Straightforward. And it is a very important step in the process. Well, in order to get some context, I would recommend you to see the first video. It was an overview of the science behind this technology, the steps necessary to produce EV OSK. Today, I will remind you briefly the content of the first video. We need some context, right? In case you have just landed here. And well, after the reminder, we will submerge again in this technology. In this video, the 401, we will learn to insert DNA inside the bacteria cells, a process that is called transformation. Okay? Transformation. And here it is, Eric Amado, that's me, ready to guide you in this field. PhD, scientist representing Novosumer, a platform to share the science behind anti-aging technologies, all that you need to understand how to stop and revert the age of the DNA, and most important, how to do it yourself. All that you need, material, equipment, steps well described, everything. Everything in novosumer.com. So let's go for the video. Let's start. Well, let's remember in a couple of minutes what we learned in the first video. We learned about the DNA, this beautiful molecule inside our cells. It contains information to shape our bodies, to build everything. Your cells read the DNA as a manual to build all their components. Okay, just remember, the DNA is just information. It's a guide to build things in our bodies. Okay, after this reminder, let me ask, do you remember something else? Maybe these little marks in the DNA? I show you that the DNA is modified with these little marks that you have here. Have a look. You see the little marks, right? The marks are stamps. They provide extra information. And sometimes they say, this DNA is old. Don't read it. It's a shame when you read it. Don't you think? When our cells see the marks, they literally read, this DNA is old. Don't read it. Well, when this happens, our cells lose functions. They get older. That is correct. As time passes, the amount of marks increase. Do you see here? This DNA contains lots of marks. As time passes, the DNA piles up a lot of these tiny marks. Well, in this DNA, the one with lots of marks, the information is no longer available, is no longer read, and our cells get older because of that, as a consequence of the marks in the DNA. That is correct. Our cells stop reading parts of our DNA. Well, it's when you get older. I'm sorry, my friend, but don't worry. Our cells have a solution for that. It's called OSK, a magic potion to revert the age of the DNA. OSK clean many of these marks. OSK polish the DNA. It reduces the amount of marks and the cells are again able to read the DNA. In other words, we rejuvenate. Oh yeah, we rejuvenate thanks to OSK. Great, right? Good to know. Well, that is the key of this technology. We provide OSK to the cells to rejuvenate them. We reactivate somehow OSK and young again. OSK, young and healthy again. Cheers! Okay, let's remember how to produce OSK. Let's go for the next slide. Okay, this is a little reminder from previous videos. This slide is a summary of the steps necessary to produce OSK. Okay, please have a look here to the left of the screen. Do you see here the DNA? This is the DNA necessary to produce OSK. So before starting with the steps, we need to design good DNA with OSK, right? This was what we learned in the second video, how to design DNA and ordering it from a DNA provider, buying it. It is easy, as you learned in the second video. You understood all the DNA involved in producing EV with OSK inside. The three DNAs, the DNA with OSK genes, the DNA with the RepCap genes, these ones are necessary to produce the EV particles, and the DNA with the helper genes, the genes that are helping to in the process. Okay, having said that, let's remember how to produce OSK, the three steps to produce OSK. Let's go for it. We just need three steps. And do you remember the first one? This one here. We produce copies of the three DNAs in bacteria cells. A lot of copies of these three DNAs. Millions of copies. Keep this in mind, okay? Many copies of these three DNAs. The bacteria cells make this work for us. We just have to insert the DNA inside bacteria cells and they will produce these copies. That is the topic of today's video, inserting DNA inside bacteria cells. Remember, this process is called transformation. It's just inserting DNA inside bacteria cells. Okay? And after inserting the DNA in bacteria, they will produce the copies for us. As you can see here, the DNA is inserted in bacteria cells and they produce many copies. And what happens next? Once we have lots of copies, we can continue to the second step, encapsulating the DNA inside EV particles. Do you remember EV? EV is the name of the innocuous virus, the inoffensive virus that encapsulates the DNA. It has been modified to help us. Well, let's continue. Are you here with me? Are you here in the second step? Let's see it. In the second step, the DNA, the three DNAs, are inserted in the second type of cells, the mammalian cells, remember. And each cell requires copies of the three DNAs. Do you see here the three DNAs with OSK, RepCap, and the helper genes? Yeah? They will work together to produce copies of the OSK genes. Do you see here? The three DNAs will work together, they will cooperate to produce copies of the OSK genes. And afterwards, the OSK genes will be included in the EV particles. These cells, the second type of cells, will do this work for us. They will read the DNA to produce copies of the OSK genes and encapsulate the copies inside EV particles. Easy, right? This is the second step. And what about the third and last step? In the third and last step, we just clean the EV particles. That's all. We purify the EV particles. We want fresh and clean particles. And that's all, my friends. All that we saw in the first video. Easy? It is simple and easy. These three steps are indeed the way it's done. It's easy, right? Clear? It is indeed. Well, as mentioned before, today's topic is the transformation. In other words, inserting DNA inside bacteria. Let's start, shall we? Let's go for the next slides. Well, here we are in today's topic finally, inserting DNA inside bacteria, a process that is called transformation. And we start with a little introduction. It is an easy concept to grasp. Look here in the middle figure. Are you here with me? On exposing cells to a dramatic increase in temperature or even a small electric shock, the membrane of the cells are transitorily disrupted. Small pores start appearing in the membrane, as you can see here. Do you see the pores here? Here, here, here. Well, this is great because we can use these pores to insert DNA inside the cells. In this case, bacteria cells. That is great, right? We can use this phenomena to insert the DNA inside the bacteria cells. Well, in Novosumer, the heat shock method has been selected. It is cheaper than the electric shock method, and the performance is similar. Plus, besides the price, the cells recover quicker from the shock. Okay, easy, right? Have a look. This slide is a summary of the process. Look here. Now to the left. You see a bacteria cell surrounded by DNA. That is what we do. We just mix DNA and bacteria cells. We mix them together. Well, have a look here now in the middle figure. You see the electric shock symbol, right? This one. And the temperature symbol. Okay? They are just simply a representation of temperature increase or a small electric shock. These are two methods to create pores in the membrane. In the membrane of the cell, the pores are the way to insert the DNA inside the cells. The DNA will pass through the pores inside the bacteria cells or other kind of cells. Easy, right? It is as simple as this. Well, what happened next? Let's have a look here to the right. The DNA is already inside the bacteria cells. In this step, we will just simply try to recover the health of the cells. We will provide a nice environment, a nice medium, a nice temperature to help the healing of the cells. The pores will disappear and the DNA will settle inside. That was easy, right? Let's go for the next slide. We will see the actual steps that are performed in the laboratory, the steps to insert the DNA inside the bacteria cells. Okay, after the introduction, let's see the steps to produce the transformation. Okay, there are only four steps, four easy steps that you can see here. 1, 2, 3, and 4 steps. And what is the first step? The first step is mixing the DNA with bacteria cells. You see it here, right? We mix the DNA and the bacteria, and we incubate the mix on ice. The temperature will be close to 4 Celsius degrees. Okay, this is the first step, just simply mixing the DNA and the bacteria. And what is the second step? Look here. Do you see here? The second step is, in fact, the heat shock, the dramatic increase in temperature. It will go from the 4 Celsius degree to 42 Celsius degree. We will expose the mix to 42 Celsius degree. It will last 30 to 45 seconds, no more. Okay? So as you can see here, we increase the temperature from 4 Celsius degrees to 42 Celsius degrees. This will create pores in the membrane, and the DNA will pass through the pores inside the bacteria cells. Okay? So now you know the heat shock creates pores, and the DNA enters inside the bacteria through the pores. That's it. Okay, let's go for the third step. Are you here with me in the third step? The third step is the recovery of the cells. We provide optimum conditions to the bacteria cells. We provide a nice temperature, a nice environment. And in this environment, the bacteria cells are able to heal and to seal the pores. You see it here. The bacteria are exposed to nice conditions and they recover, they heal, they are in nice conditions. So this is the third step. And what about the fourth step? We plate. Whatever we have here, we plate into plates with a solid medium, with a medium with agar. It's kind of a gel that is very nutritive. Okay, so the fourth step, as you can see here, is just plating. It's just seeding the plates with the sample that we recover from here. So we seed nutritive agar plates with bacteria and DNA inside. The bacteria cells will grow in the surface of the agar plates. So that is to say, the bacteria will grow here in the surface of the medium with agar. Okay, easy, straightforward. Uh, it was indeed. So let's go to see the step by step in more detail. Okay, let's go for the next slide. So we are in the first step. Mix the DNA and the bacteria cells and incubate them. Okay, so what do we need? First, we need a nice ice box like this. Okay, an ice box that guarantees around 2 to 4 Celsius degrees temperature. And what do we need? We need a vial of cells with cells, competent cells. Competent cells are cells that are chemically prepared to accept DNA inside. Okay, so here you see competent cells. What do we need? What is the other thing that we need? The DNA, right? So here we have another vial of another vial with DNA is here. Okay. Okay, so we have the competent cells and we have the DNA. We have to thaw them. So we thaw them. Little flicks will help it, okay, to thaw the DNA and the competent cells. And what do we need? Again, we need another Eppendorf tube. An Eppendorf that is sterile and empty. We place it on ice as well to reach a temperature near or close to 4 Celsius degrees. What is the next step? The next step is transferring the DNA, the DNA and the competent cells together into the Eppendorf tube. Here we are. We use a flame to guarantee a sterile atmosphere. The flame will guarantee more or less 20 to 25 centimeters of sterility. So as long as we work near here, we will be ensuring that the atmosphere is sterile. So the only thing that we have to do is transferring 25 to 50 microliters of these cells into the empty Eppendorf. And afterwards, we will add 100 nanograms of DNA, this DNA, into the same Eppendorf tube. Okay, at that point, this Eppendorf tube will be filled with the DNA and the bacteria cells. So just close the Eppendorf and transfer it back again to the ice box. So we have the Eppendorf here with the DNA and the bacteria. The only thing that we have to do is incubate this DNA with the bacteria for, let's say, 5 to 30 minutes on ice. And that's all, my friends. That's all that we have to do in this step. Let's go for the next step. That is the heat shock. That means increasing the temperature a little bit drastically. Okay, let's go for it. Well, we are in the second step, the heat shock, increasing the temperature dramatically to create pores in the cells and insert the DNA through these pores inside the bacteria cells. Here we are. As you can see, we are going to increase the temperature to create pores and include the DNA inside the bacteria cells. Okay, so let's go for it. What do we need first? Remember, we had an Eppendorf tube with the DNA and the bacteria cells. Okay, here it is. So what do we need in this step? In this step, we need a floater. A floater is a piece of plastic that floats above the water. Do you see here? So our cells are here at 4 Celsius degrees, and we have put the tube inside a floater that will float above the water. So what is the next step? For the next step, we need a timer. The time is very important, so we need to measure the time. Okay? And what else do we need? We need a water bath. This water bath is set up for 42 Celsius degrees, as you can see here. This is a water bath. This is the representation of a water bath. You see here, and above the water bath, you can see the Eppendorf floating thanks to the floater. So the second step is just transferring the Eppendorf with the floater to the water bath. We transfer it and we start the timer. After 30 to 45 seconds, careful with the time, only 30 to 45 seconds, we take the Eppendorf and we transfer it again back to the ice. Okay? So in this second step is the important one. This dramatic change in temperature from 4 Celsius degrees to 44 Celsius degrees is responsible for the creation of pores in the membrane. Remember. So that's what we have done. We expose the cells to 45 seconds at 42 Celsius degrees, and afterwards we return the Eppendorf to the ice. The Eppendorf with the cells with pores and the DNA will rest here for less than 5 minutes. Remember, less than 5 minutes. After these 5 minutes, as you can see in the next slide, we will provide nice conditions, a nice environment for the cells to heal, to recover. That will be the content of the next slide. Okay, easy? Let's go for the next slide. Okay, let's go for the third step, the recovery of the cells, which is equal to sealing the pores, healing the cells. This is the third step, is just making the cells happy again, leaving them in a nice environment to heal. Okay? And a nice environment for them to heal is an environment with a rich medium. That is a medium with a lot of nutrients, a lot of food that they like at 37 Celsius degrees. Okay? 37 Celsius degrees as the human body. 200 RPMs agitation. That means we create some movement, some agitation. So they are going to be in a smooth movement of 200 RPMs. And we will leave the cells for 1 to 2 hours. Okay? So what do we need for this purpose? Okay, remember we had the cells with the DNA inside on ice. Okay? And what we need is, remember, the rich medium, the medium with food. This medium, this rich medium, is pre-warm. Is pre-warm, as you can see here, in a warming chamber, and is pre-warm at 37 Celsius degrees. So we have everything that we need now. We have the cells with the DNA inside, and we have the pre-warm medium. What do we have to do? Is just mixing them together in an stereo environment. Okay, what do we need for mixing it? We need an stereo environment thanks to the flame, and we need to transfer the medium to the cells with the DNA inside. We use a pipette for this purpose. Okay, so the only thing that we do is transferring 200 microliters of rich medium. I repeat, 200 microliters of rich medium to the cells, and we mix a little bit, maybe flicking the Eppendorf tube or maybe using the pipette very gently. So that's this step, adding the 200 microliters of rich medium to the cells and mixing them together very gently. As you can see here, this is a step. Okay, in the picture, it's nice to see the things in a picture, right? Okay, and the last step is just providing the rest of the conditions for the cells. We want the cells to be happy. Okay, so what we need is a warming chamber, an incubation chamber. It looks like this. These chambers provide provides a steady temperature of 37 Celsius degrees and also provide agitation. Let's see let's see this picture here. It represents this chamber. Inside these chambers, there is a motor here in the base that agitates whatever is inside. And we set the chamber for 200 RPMs. It's a gently agitation. So what we have to do is placing the ependor tube with the cells, with DNA, and with the rich medium inside the chamber. We put the ependor inside the chamber and we leave it for 1 hour in agitation and at 37 Celsius degrees. These conditions that we provide to the cells are enough to make the cells happy, so they will heal, they will recover, they will seal the pores. Okay, this is this step. Let's go for the next and last step. Plating the cells is just plating the cells. Okay, let's go for it. We are in the step number 4, seeding bacteria cells into nutritive agar plates. Okay, we will seed bacteria cells in the surface of these agar plates. This nutritive medium that is solid, is kind of a gel. Okay, so this step number 4, in this step number 4, we seed nutritive agar plates with bacteria cells that have the DNA inside. Do you see here? This is a representation of an agar plate. The actual agar plate look like this. These are 2 agar plates that are going to be seed. And what do we need for this purpose? Well, first we need the ependor tube with the bacteria cells and the DNA that is inside the bacteria, and we also need the agar plates, the LB agar plates. These plates with medium that is solid, kind of a gel, and have good conditions for the cells to grow. Okay, so once that we have the cells, the recovered cells, and the agar plates, what do we do? Well, we do as I'm going to describe to you. Well, remember, we need to work under sterile conditions with the flame, right? Okay. This step, it's just transferring 60 microliters of these recovered cells. It's just transferring these 60 microliters of recovered cells into an agar plate. Okay, as you can see here. And we will use for this purpose a nice pipette. Well, you have a picture here, a real picture with the pipette that has in the tip 60 microliters of cells, and they are transferred to the agar plates, as you can see here. And what do we do next? Okay, this step again under sterile conditions with the flame. For this purpose, we use this thing here. This tool here is just a piece of plastic, sterile, that help us to spread the cells, the 60 microliters of cells. So the only thing that we have to do is kind of sliding this tool along the surface of the agar plates. By doing so, what we are doing is spreading the bacteria cells. Here you have a nice picture with the agar plates that have the 60 microliters on the surface, and this plastic tool is helping us to spread the bacteria cells on the surface. Well, once we have all the bacteria cells nicely spread over the surface, The only thing that we have to do is incubating these agar plates into an incubation chamber. Do you remember the incubation chamber? Okay. This incubation chamber does not have agitation. We don't require agitation for this purpose. The only thing that we need is this chamber being able to hold a temperature of 37 Celsius degrees. That's all that we need. A steady temperature of 37 Celsius degrees. So we will put this plate inside the incubation chamber at 37 Celsius degrees, and we will incubate it for 16 to 24 hours. Okay, here you have a nice picture of the incubation chamber set up at 37 Celsius degree with the agar plates inside. The agar plates with the bacteria cells, with the DNA inside. Happy? Easy? Okay. I hope you are, because the process has finished. After this, after 16 to 24 hours, we will get our results. Let's go for the results. Let's have a look on the results. Okay, so finally we got our results. After 16 to 24 hours, we have our results. What are these results? The results are colonies of bacteria with DNA inserted, colonies of bacteria in the surface of the agar plates. The results are very much like this. Do you see here the picture? Do you see these little dots? These little dots are colonies of bacteria, millions of bacteria in each colony. Here you have a representation of an agar plate with colonies on the surface. The colonies will grow on the surface. And let's read with me, okay? Because it says here, only bacteria with cells with DNA insert will survive, will grow, and create colonies. Okay, so what is this telling us? It's telling us that there were bacteria with the DNA, but there was bacteria without the DNA, because the performance of the process is not 100%. So, okay, only bacteria with the DNA is able to grow. Only the bacteria with the DNA insert grow. So how do we do it? How did we do it? Okay, I didn't mention that these agar plates, in fact, LB agar plates, have antibiotics inside. Okay, it has antibiotics inside, and the antibiotics, as you know, kill bacteria. So, okay, so we have agar plate with antibiotic that kill bacteria. How is it possible that these bacteria grow? Okay. Do you remember the three DNAs? The OSK DNA, the RepCap DNA, and the helper DNA? In these three plasmids, we have a region that is called the resistance gene. The resistance gene, as you can see here. So this name is very descriptive. This name is telling us that this piece of information is able to help the bacteria to survive the antibiotic. By having this region inside the DNA, the bacteria will be able to survive the antibiotics. It's an easy concept, right? So now we know that only the bacteria with the DNA inside is able to grow. Easy? Remember, the three DNAs have as well a piece of DNA, a region of DNA that enable them to survive the presence of antibiotics. And that's why only bacteria with the DNA will grow, as you can see here in this nice picture and in this nice figure representation. Okay, so that's all, my friends. These agar plates with the bacteria on the surface can be kept at 4 Celsius degree in a fridge. So we keep these agar plates with bacteria in fridges at 4 Celsius degrees up to 10 to 14 days. After 10 or 14 days, we will have to discard these plates. Okay, easy? Let's go for the next slide, the summary of what we have just seen in this session. Well, we are wrapping up, and we cannot say goodbye without the summary of what we have just seen in this session. So we perform 4 steps. Just 4 steps are necessary for transforming DNA inside bacteria cells. What we did first was mixing the DNA with bacteria cells, as you can see here in the figure. The second step was the heat shock, a dramatic increase in temperature. It went from 4 Celsius degree to 42 Celsius degrees. This dramatic change in the temperature create pores in the surface, in the membrane of the cells. And these pores are used by the DNA to enter inside the cell. Once we have the DNA inside of the cells, the third step is recovery. The recovery, the healing of the cells, the sealing of the pores. We need the cells healthy. Okay, so what we do in these steps is just providing to the cells optimum conditions to heal. And after healing, the fourth step is just plating, is seeding the cells in plates and give them optimum condition, this time to grow. Okay, so in the third step, they recover, they heal, and in the fourth step, they grow as colonies, as you saw in the previous picture. And these colonies have the DNA that we want inside. Easy? It is easy. Well, I have to say that this information is well detailed in the form of protocols, exhaustive protocols. These protocols have all the information, providers, material, uh, full description, because you have to understand that this is just a presentation. This has key points to understand the technology, but all the technology is in the website that will be available in the following months. Okay, you will find this information in the form of a protocol, SOP, or working instruction, that is called 32, blah, 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 plasmid amplification, step 1, transformation. You will find this protocol, I'm sorry for the blah, 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 in the website. Okay? I have to mention as well that this protocol is compliant to CGMP, ISO 9001, ISO 45001, ISO 14001. All of them high standard quality protocols, environmental protocols, health and safety protocols that ensures that the process is suitable for industry, is suitable for humans. Well, I have said there will be ready and available for free in novosumer.com in the following months. And now we are ready to say goodbye. Thank you very much. It's been my pleasure again to be with you, guiding you into this world of gene therapy, specifically in DNA rejuvenation. Every time, every single session is a pleasure. I hope this information resonates with you. Don't you think, Novosumer? Novosumer wants to say thank you million times to you to share this path, to share this time with us. Thank you very much. Have a lovely day. Enjoy, and I hope I see you in the next sessions, in the next videos. I hope you are there enjoying them, learning, or maybe creating yourself this technology, implementing in your industry. Thank you very much. Bye bye.